This man explains how scientists managed to simulate a mouse's brain.
This is really remarkable neuroscience news. Scientists have essentially simulated a part of a mouse's brain. They had the mouse watch a bunch of videos, and then they imaged all of its neurons and all of its neuronal activity. And then the scientists took out the visual cortex, a piece of it about the size of a grain of sand, a cubic millimeter, and then they sliced it into 28,000 different slices using an electron microscope. Combining the images from the neurons and how they were acting and the images from the actual structure, they made a digital map of the brain using AI, because this would take forever if they did it by hand. They used AI to predict how the neuron's activity would match up with the structure and the surrounding neurons. So they created a gigantic map of a fraction of the visual cortex of the mouse. In total, it's about 500 million synapses, 200,000 neurons, and about 1.6 petabytes of data. That's about 1.6 million gigabytes. Now, using this fully completed simulation of a part of the mouse's brain, we can determine how the neurons would behave when we give it certain stimulation, like when we show it a movie or we show it visuals or shapes or things or colors. We can now predict which neurons will fire off in the mouse's brain. So now for the first time, we can actually see how the structure of the brain directly gives rise to its function. In 1979, a biologist called Francis Crick, who was a Nobel Prize winner, he said that this would be impossible. He said that if we take a cubic millimeter of brain, you'll never be able to actually understand all of its activity. Well, now we've done the impossible. And what this means for humans is that eventually maybe we'll be able to determine how our neurons and our exact brain structures give rise to how we think and how we feel and all of our perceptions. And this might be able to help us fix certain psychological disorders in the future. Imagine one day having a complete map of the human brain and all of its neurons and all of its synapses, and being able to determine how conditions like autism and schizophrenia and depression and anxiety and Alzheimer's, imagine understanding how those things occur and develop and how to stop them and how to prevent them and how to fix them. Imagine going into the brain and going directly towards those circuits in the brain that give rise to those conditions and fixing them like that. That's what's possible with this kind of understanding. These next 20 years of neuroscience are going to be so enlightening for us as a species.
Alright y'all, this one's coming hot off the press. Yesterday, a group of scientists reported for the first time in human history, DNA editing has been used in vivo to save a human baby's life. Let's talk about it. There are two ways that DNA editing-based therapies can happen. One of them is called ex vivo, or outside of life. Basically, you take cells from a patient, fix the mutation that's making them sick, and then take those tweaked cells and put them back in the patient's body. This was the approach used in the world's first CRISPR-based therapy, Casgevy. Doctors take bone marrow cells out of patients with sickle cell anemia, tweak the mutation that's making them sick, and then put those bone marrow cells back in so that they can start cranking out healthy red blood cells. But there's another way to do this called in vivo, or inside a living body. Basically, you take the DNA editing machinery itself and inject it directly into a human patient, so all of the editing occurs inside of their body. And for the first time in human history, we've done it. A baby was born with a mutation that causes a disease called CPS1 deficiency. This is a lethal illness that basically turns protein into poison, but you still need to eat protein in order to survive. That's why CPS1 deficiency carries a 50% mortality rate in early infancy. Within just 48 hours, this baby was showing symptoms that were threatening his life. Doctors predicted that he would need a liver transplant before he was even 5 months old, but little tiny livers are very hard to come by, and the transplant process is extremely dangerous in a baby that's already so sick. So his parents did something incredibly brave. They allowed a team of scientists at Philadelphia Children's Hospital to try out a new in vivo DNA editing therapy to try and save their son's life. The scientists used a flavor of CRISPR called base editing. Basically, your DNA is made up of two strands that are connected in a double helix. Original flavor CRISPR breaks both of those strands, but base editing only has to break one of them, which means it's a lot gentler on your DNA. This amazing team of researchers, including one of the inventors of base editing, an evolution hacker who I made a whole video about not too long ago, injected DNA editing machinery directly into the sick infant. Within 7 weeks, doctors were able to increase the amount of protein that the baby was able to eat, halve the dosage of the medication that previously had been keeping him alive, and establish that he showed no adverse effects to receiving the treatment. Now, this is all very preliminary. This is only done in one patient and very recently, so there's a lot of work that still needs to be done, both assessing the long-term effects of the treatment on this one patient and increasing the number of patients that have received it. But this is a huge deal. For the first time in history, we have edited a person's DNA inside of their body, rather than in cells in a dish that later get transplanted back into them. And so far, it looks like it may have saved that human being's life.
This woman breaks down 5 mind-blowing facts about sharks I can bet you didn't know.
I bet I know more about sharks than you do. I'm going to give you 5 shark facts. And if you didn't know all 5 of the shark facts I'm about to tell you, which you probably didn't, you have to share my post. Deal? Deal. Let's go. Number 1. Many sharks start hunting before they're ever even born. The egg will hatch inside of the mom, and the shark will eat its siblings before it's ever even birthed. Number 2. There's a lot of sharks who only eat other sharks. No fish, no crustaceans, no whales, other sharks. Number 3, in the year 2013, they did a study on bamboo sharks and found that sharks, before they even come out of the egg, can sense danger. Number 4, they found hammerheads and silky sharks living inside of an active volcano, meaning it regularly erupts. And their evolutionary adaptations allow them to live there, and we don't fully understand that. Number 5, sharks were able to solve puzzles simply by watching other sharks solve them. So they're pretty smart.
This man claims trees don't really exist and gives some really solid facts why.
Scientifically speaking, there is no such thing as a tree. They just don't exist. I'm not saying when you go outside and you see a tree that your eyes are deceiving you, but what I am saying is that from a scientific standpoint, they're not really a concrete separate thing. Basically, if you've taken a high enough science class, you're probably familiar with this, a phylogenetic tree. And you're probably aware of the process of speciation. So knowing that, you would look at a phylogenetic tree and expect to see a specific branch for trees, but that isn't there. And that's because trees aren't their own separate species of plant. There are a bunch of different types of plants that have all evolved in a really similar way. You see, plants are constantly racing with one another to see who can collect the most sunlight, because they need that for photosynthesis to have energy. And one of the best ways to get more sunlight than everyone else is to be taller. And to have more leaves, so that way you have more places for sunlight to be collected and for photosynthesis to actually begin. Your regular old run-of-the-mill plant stock can only get so tall and support so much weight before it begins to fail to support the plant. It'll start to bend towards the ground or even break, which is obviously not good for the plant's survival. But bark is much stronger and better able to support larger plants, allowing them to collect more sunlight. On top of that, wood is really good at transporting water throughout the plant and at storing sugar, all of which allow the tree to justify its much larger size. A bunch of different species of plants all throughout the world all realized that this would be advantageous, and so they developed this independently of each other. This is convergent evolution, where the same characteristic is developed independently in different organisms, which can lead you to mistakenly believe that these characteristics are holdovers from a common ancestor. In reality, these traits or behaviors develop due to the fact that they're facing similar environmental pressures. It's why this bird was able to just re-evolve. In reality, there were 2 species of this bird, one of which was flightless and was wiped out when the island that they were living on was flooded. The species with the ability to fly then repopulated the island and over time became flightless as well, re-evolving basically into the same bird that was wiped out before, because the pressures from their environment were so similar that they basically just turned into the same bird. But yeah, that's why trees don't really exist, and some other organisms don't really have time for that.